You're listening to Conferences Online Allergy from Children's Mercy Hospitals and Clinics in Kansas City, Missouri. Today is July 9, 2012, and I'm your host, Dr. Jay Portnoy. Our topic today, laboratory tests for immunodeficiency. Our presenter is Dr. Thomas Fleisher. He's the chief of the Department of Laboratory Medicine at the National Institutes of Health in Bethesda, Maryland. And I'll introduce our first presenter, Dr. Tom Fleisher. Dr. Fleisher is the chief of the Immunology Services Department of Laboratory Medicine at the National Institute of Health in Bethesda, Maryland. You can see his biographical sketch below. Dr. Fleisher has been instrumental in teaching us about laboratory testing. If you go to any of the national meetings in allergy, he'll almost certainly be on the plenary session talking about the latest tests. He's in charge of the laboratory tests at the NIH, so obviously he's clearly one of the at the forefront of laboratory testing. So we really appreciate your willingness to join us today, Dr. Fleisher. Here are your slides, and if you'd like, I can give you control of them, and that way you can let me just give you that. Dr. Tom Fleisher, you've now got control, so if you move your mouse, you'll be able to control the slides. There you go.
Okay.
So thank you for joining us.
Okay, Jay. So we'll just sort of walk our way through. For some of this, it'll be review. Hopefully, there'll be some new information. So always important to remember that we're all clinicians. I'm actually not a pathologist. I was trained in pediatrics and then A&I. So the medical history is always the important starting point when you're thinking about any sort of a defect in terms of post-defense. Obviously, many of the diseases we deal with are congenital disorders of immune function, so a careful family history, particularly paying attention to early deaths, history of recurrent infections, etc. Physical exam, to be frank with you, is sometimes revealing, but in many cases does not provide specific clues with regard to where we need to go. It's really the history that is the driving force. And since we now have close to 200 genetic defects associated with immune dysfunction, and we're becoming much more aware of the fact that a particular genetic defect can actually be associated with a wide range of clinical phenotypes, we have to keep our minds open in terms of diagnoses, potential underlying defects, but ultimately the medical history directs us with regard to what type of immunologic evaluation we need to undertake. By and large, it's expensive testing, and so shotgun sorts of approaches are really discouraged. So I'm going to go through using the traditional sort of organization of the immune system, but there's a caveat that we ought to all be aware of, and that is that none of the various arms of the immune system work in isolation. There's a substantial amount of overlap, of redundancy, and cooperation. So for the sake of discussion, it's very useful to talk about each of these arms as an independent entity, but keep in mind that the immune system is in fact a very cooperative system. So if we're talking about B-cell function or antibody production, clearly the clinical history is one that's quite distinctive, and that's of recurrent sinopulmonary infections with high-grade encapsulated bacteria, Haemophilus influenzae and strep pneumoniae being the classic examples. I think all of you are aware the screening tests start with immunoglobulin levels. We probably should include IgE because there are a number of disorders that are in fact associated with elevated IgE in the face of abnormal antibody production. Specific antibody responses are the next step, and clearly we distinguish between protein antigens and carbohydrate antigens because the immune system sees them fundamentally differently. And in addition, the maturation of the immune system relative to responses to protein antigens versus carbohydrate antigens is different. Finally, HIV testing ought not to be automatically excluded in somebody with a history that is somewhat more suggestive of antibody deficiency, particularly in the pediatric population, although we're all well aware of the fact that patients with HIV-related immunodeficiencies primarily present with defects related to T-cell function rather than B-cell function. If we talk about immunoglobulin levels, you are at a children's hospital. I hope that this is speaking to the choir, and that is that age-related differences are significant and must be considered for each evaluation. The data has to be looked at in the context of age-appropriate reference intervals. That really is true for most of the immune function testing that we do. Remember also that a laboratory typically provides you 95% confidence intervals. That means that 2.5% of the population evaluated to generate this range were above and 2.5% were below. So being below is not automatically associated with abnormal immune function. I also should just as an aside point out
just a question, if it's a skewed distribution, couldn't like 4% be below and 1% be above, something like that?
No, not when you do it 95% confidence interval.
It's always symmetrical.
Above and below. The only caveat would be if you are looking at something where 95% of the population is below. So for instance, an ANA level, 5% of the population would be expected to be positive using a 95% confidence interval. There's no way you have too low of an ANA. But for globulin levels, it's a central 95%. And in fact, it's inappropriate to talk about a mean IgG level. They do not distribute arithmetically. They distribute geometrically. For you who are computer-based, you don't even know about logarithmic transformation, talking about geometric means and times divide. So we'll just ignore it. 95% confidence interval is not a Gaussian distribution using real numbers. So put that in the back of your mind. Sorry. Half-life I've given you here only to point out that there is a difference, and clearly IgG has the longest half-life, IgE the shortest. Also keep in mind that immunoglobulins, as is the case for all proteins, are actually the combination of the net of production, consumption, and loss. So not all low immunoglobulin levels are a production problem. Sometimes it's actually a loss situation, so you can have GI protein loss. But the important point to keep in mind is that if you're losing immunoglobulins, you're also losing albumin. And so the key to help differentiate between loss and production is look at an albumin level. If it's normal, you cannot subscribe to increased loss. So that is a nice test point. There are a couple of diseases in which there is hypercatabolism of immunoglobulins. They're very uncommon, and myotonic dystrophy is one of them. It's really only a test point. It's not going to come up in your evaluation of patients. So the specific antibody responses, the follow-up really is the in vivo test of how the immune system is able to make specific antibody. And so we talk about natural antibody screens. So you can test for isohemagglutinin if your blood bank is willing to do that. That's obviously anti-A or anti-B. That is obviously not of utility in somebody who is of AB blood type, but that's quite rare. It's also not very useful in a young infant. The other thing that one can do if you have a very careful and accurate history of immunization is to just do a random level for antibodies to prior immunizations. The difficulty there is obtaining very accurate information about prior immunogens. What typically is recommended in this setting is provocative testing, immunizing with both proteins, so tetanus toxoid, diphtheria toxoid, and carbohydrate antigens, keeping in mind that the conjugates that you all use in pediatrics do not truly test the capacity of the immune system to respond to carbohydrate antigens because they're conjugated to proteins And so, if you will, they're tricking the immune system or bypassing the standard response to carbohydrate antigens. And so Pneumovax is really the true test of a response to carbohydrate antigens. Typically, one would look at a pre- and then 3 to 4 week post-immunization response. And I have to be honest and tell you that what constitutes a normal response to Pneumovax is variable. It's dependent on the beholder. So most people would say if you respond to 8 of 12 serotypes, that's entirely normal. What do you do with 6 of 12? It's not really abnormal. So this is one of those areas in immune evaluation where absence of response is a slam dunk, simple. Full response is simple. And the shades of gray become a little bit more complex. And you will see varying interpretations in terms of what is truly abnormal. And so this is a particular issue in people that have normal antibody levels and have what is called specific antibody deficiency, SAD, which is debated in terms of how that diagnosis is established. And we don't really have time to go into all of the fine points of the debate. And Dr. Portnoy will guide you in what is right. So IgG subclasses, I just like to put in my 2 cents worth. The test is very expensive. Subnormal IgG subclass levels alone appear to have little clinical significance. And typically you must follow up with functional antibody response to immunization if you're going to justify going to replacement therapy. So I actually wonder why you order the subclasses. I mean, why not just go to specific antibody responses and circumvent a $125 or $150 test? To my mind, it just doesn't really help very much. Now, one caveat may be in an IgA deficient patient who really is having significant problems with recurrent infections, that really has a history that's strikingly abnormal. And we know that IgA deficiency in general is not clinically a terribly significant diagnosis in terms of recurrent infections. But we also know that there's a spectrum and that IgA deficiency may be associated with either an emerging process that may ultimately look like common variable immunodeficiency, or may be accompanied by subclass deficiency, so it's more complex than just selective IgA deficiency. And those are the patients who are most likely going to have serious recurrent infections. And we worry about that because these recurrent infections can lead to bronchiectasis and ultimately can, if not treated aggressively, can result in pulmonary failure and death. So a patient with a history of recurrent pneumonias that are diagnosed based on X-ray findings, this is a patient who really is at risk for developing serious pulmonary disease that can really interfere with life expectancy. Okay, so if we switch gears now, talk about T-cell dysfunction. Again, reminding you that it's an artificial distinction. We know that patients that have significant T-cell abnormalities have a history that is different than patients that have purely antibody deficiency. They have recurrent opportunistic infections. They get pneumocystis. They get CMV. They get fungal infections. I mean, it is a group of intracellular pathogens that is strikingly different than a pure antibody deficiency. When we talk about screening tests, we really have much less to offer. We can do a lymphocyte, absolute lymphocyte count, which you can calculate from a CBC with a differential. The reason this is of utility is that 75%, give or take, of the circulating lymphocytes are T cells. So if you're dealing with a defect in which there is either a developmental block in T cell production or a process such as HIV that results in destruction of T cells, you would expect to see lymphopenia. If you have no B cells, which represent 10 to 15% of your total lymphocytes, that will not be a significant impact in terms of the overall total lymphocyte count. The other thing to keep in mind is that we use in adults 1,000 as the cutoff for lymphopenia and 500 for severe lymphopenia. In an infant, the cutoff is 3,000. Again, pointing out that age-related reference intervals are absolutely required to properly interpret data when dealing with children. Delayed type hypersensitivity testing I'll touch on a little bit more. It's really being used less and less. And in this setting, an HIV test is an absolute necessity. Keep in mind that if there's any question about antibody production, then doing an ELISA and a Western blot is of no utility. And you really need to go to a nucleotide-based, a PCR-based assay for HIV. If you can't make an antibody response, obviously doing a test for HIV antibody is really nonsense. For some reason, all right, well, it's not wanting to move. Oh, okay. Sorry. A little bit of a lag here. So DTH testing, just to point out, you do it for PPD. Think about what the test involves. You're actually taking antigen-specific T cells, that is previously exposed memory T cells, that are activated. They secrete cytokines, and then inflammatory cells respond to these cytokines and chemokines, migrate to that local site, and induce an inflammatory response over 48 to 72 hours. So it's actually quite a remarkable chain of events induced by exposure to a recall antigen. Historically, we have used a variety of recall antigens such as tetanus toxoid, Candida albicans, mumps antigen, as well as a PPD. And so you really had a nice in vivo test that would be examined 48 to 72 hours after exposure. The one caveat being that lack of response either means lack of prior exposure or anergy. Hence, the use of multiple recall antigens to try to rule out that lack of exposure is the basis of no response. Unfortunately, there's not a big market for these products. Obviously, tetanus toxoid is available. I'm told that the Candida antigen is becoming less and less available. And so, by and large, people are not using this particularly frequently. I don't know if you use it at Mercy or not.
We don't have access to them. I think the only one that's readily available is Candida, but you'd have to get it from an allergy extract company as an intradermal product.
Right.
So we have that.
By and large, this has become of historical interest. Too bad, because it is a very inexpensive screening test that does provide some utility. And many years ago in HIV, actually, there were a number of studies that demonstrated that it had great predictive value in terms of outcome, somewhat equivalent to CD4 cells. So really, we're stuck then, if you look at a lymphocyte count, we have to go to in vitro testing. And so I'd like to just point out that there are two general categories in terms of in vitro assays that one would use to further explore T cell or cellular immune function. One involves immune cell identification. Flow cytometry is the standard fare. And the other is immune cell function. And clearly, this is now getting into high-cost testing, so it really needs to be ordered based on a strong history of a cellular or T-cell immune defect. And there are assays, T-cell proliferation assays, cytokine assays. I'll come back to these in just a moment and briefly discuss. The one T-cell effector assay that's available is a cytotoxicity assay. They're not generally available in most labs. There are some specific settings where it's very useful. And along those lines, not truly a cellular immune in the sense of a T-cell, but rather in the context of cytotoxicity is the natural killer cell cytotoxicity assay, and that's particularly relevant in settings of HLH. So if we talk about immune cell identification, we're really talking about cell types and subtypes, and it depends on identifying cell surface protein expression using a host of monoclonal antibodies. I hope you all know the CD nomenclature, at least at its outset. We're now well above 300 in terms of cell surface proteins, not only focused on lymphocytes, they're also present on myeloid cells, present on platelets, they're present on neural tissue. So it also points out this incredibly complex landscape on the surface of cells utilized to generate signals to the cell. Enumeration depends on having a flow cytometer and labeled monoclonal antibodies, labeled meaning fluorochromes. These are chemicals that are excited by a particular wavelength of light, emitting a signal of lower energy, meaning of higher wavelength, that can be detected with a photodetector. And we call this process immunophenotyping. Remember, counting cells is not the equivalent of function testing. And even when we look at specific cell surface markers that have functional equivalence, it is still a counting system. And so I like to use the analogy, if you're in an airplane and you're flying at 30,000 feet and you look down and see a large factory, you know the factory is present, the building is there. What you don't know is whether the factory is actually in production. And so we know the cells are present when they have characteristic markers. What we don't know is whether they're fully functional. And that really depends on doing functional testing. This comes up currently quite frequently when people talk about T regulatory cells. They always talk as if identifying them by flow is the equivalent of assaying function. That is not the case. All right. So how do we use immunophenotyping? Well, we can use it as a diagnostic tool to evaluate for the absence of a lymphocyte population or subpopulation. I'll give you a couple examples in just a moment. You can evaluate for specific cell surface marker, that is a specific protein linked to a specific disease. So I picked CD40 ligand, the protein that is defective in X-linked hyper-IgM syndrome, also known as CD154. I put in there a caveat. This is not a constitutively expressed cell surface protein. You have to activate T cells first in order to see its expression. So if you look in peripheral blood of unactivated cells, nobody will express CD154. You first have to activate them ex vivo and then look at the cells after 24 hours. In contrast, the interferon gamma receptor, which is actually expressed on monocytes, and we know there are multiple defects with this receptor, that's a constitutively expressed receptor. You can stain the monocytes immediately after a blood draw and look for the presence of a receptor. So that really sets the stage. When you're looking at specific protein, you have to understand the biology. Is this constitutively expressed, or does it require prior activation to be expressed? And obviously that dictates how you look at it. Assessment for a biological effect tends not to be diagnostic, but you can look for B-cell isotype switching, looking at surface immunoglobulin production. You can look at memory T cells and or B cells. You can look at naive T cells. So there are a number of things that one can do, and there are a number of other things in which biological effects of immunologic dysfunction can actually be examined by flow cytometry. Again, as a surrogate of function, but not an actual test of function. So here is a very, very simple flow dot plot. And you can see here is a flow cytometric evaluation of a patient with hypo or AGAM. Let's make this an infant boy, no B cells, plenty of T cells. This is X-linked agammaglobulinemia. You go on to evaluate BTK by mutation analysis, you confirm it. But keeping in mind, if it's a girl, unless she has abnormal lionization or has Turner syndrome, it's not going to be XLA, right? Because 2 X chromosomes make somebody a carrier. I've never heard of anybody inheriting 2 defective X chromosomes for the BTK gene. So we know there are autosomal recessive A-gamma conditions. There's at least 4 different genetic defects. If this was an adult with absolute absence of B cells, the first thing that should cross your mind is A-gamma with thymoma or Good syndrome. This typically is late middle age or older. You can see it rarely in common variable immunodeficiency, but that's very atypical. They usually have B cells. Okay, change gears, very different picture. I've tried to highlight what we're looking at, no NK cells. Okay, there look like there's some dots for T cells, but that's about 15%. This is a child with a total lymphocyte count of 600. Remember, an infant. Remember what I said, 3,000. So you're talking about 90 T cells? Pretty low. Above 2,000 is typical for an infant. But the B cells are entirely normal. 500 plus B cells. So what is this? T minus B plus NK minus SCID. And so you can see the 4 different flavors outlined. Absence of T cells, B cells, and NK cells. That would be most classically ADA minus or ADA deficiency. Absence of T cells and B cells with the presence of NK cells. What's the common thread there? Both T cells and B cells have an antigen receptor. If you can't make an antigen receptor, you can't make T cells, you can't make B cells. So what defects cause the absence of the capacity to make an antigen receptor? So the recombinase activating gene, or RAG defects, RAG1 and 2, Artemis, ligase 4, etc. One major point, and I hope there's an immunodeficiency lecture that follows, RAG1 and RAG2 are unique to the immune system. Remember, you have to be able to do a somatic, you have to recombine various genetic elements to generate a mature antigen receptor, both the T cells and the B cells. This requires RAG function. That's unique to the immune system. So the only impact of RAG deficiency is on immune function. Artemis, ligase 4, these are all involved not just in the immune system's recombination process, but they're actually DNA repair enzymes, and we are all subjected continually to damage to our DNA. So patients that have these defects have increased radiation sensitivity. That's the classic. They also may have other findings, microcephaly and some other sort of clinically unique findings. But the fact of the matter is that because these are DNA repair enzymes, they have a particular sensitivity to radiation. And if they develop cancer, this becomes a major issue. It's actually even an issue just for doing CTs. T minus, B plus, NK minus, the most common, the picture I showed above. X-linked SCID, a defect in the common gamma chain of the interleukin 2, interleukin 4, interleukin 7, there's 5 different cytokine receptors, or in JAK3, the autosomal recessive equivalent to the common gamma chain defect. And finally, a defect that only impacts T cell development, but impacts B cell function as well, and that's classically the IL-7 receptor alpha chain. It tells us that IL-7 is an absolute requirement for normal T cell development. So that's enough said about SCID. I hope you'll hear more. You know that it presents early in childhood. And we're going to come back and touch on newborn screening, which is really one of the major new efforts going on. So if we turn from phenotyping, I didn't do anything about biological effects. We're really pressed for time today. Lymphocyte function testing, it's in some levels, for instance, in the SCID setting, it really is an absolute necessity. And so we typically, and this has been the case now for 40 years, I was doing PHA responses in the early 70s. It's proliferation, a response to mitogens, recall antigens, which in many ways would be the complement to delay type hypersensitivity response. A response to a recall antigen requires a memory T cell that has been previously exposed to that recall antigen. One can use, for instance, tetanus toxoid or Candida albicans as recall antigens in vitro. And one can use allogeneic cells. So these are cells that are disparate in terms of HLA class II antigens, DR, DP, and DQ. The typical way that proliferation has been done for decades is based on tritiated thymidine incorporation. Thymidine is one of the nucleotides incorporated into DNA, and so when you add tritium, you can then count this using something called a scintillation counter. Tritium is a beta emitter, and so any cell that has incorporated thymidine, because it is undergoing DNA synthesis as part of its proliferative process, will actually generate a light signal in a scintillation counter. And so you end up with results that are most easily interpreted based on counts per minute. So you put a fixed number of cells and put it into a scintillation counter with cocktail. Alternatively, you can look for cytokine release into supernatants, gamma interferon, etc. There are also flow cytometric methods that can be used to guide you in terms of T-cell proliferation. There's something called cell tracking dyes that look at the number of divisions in response to a proliferative signal. There are assays that look at activation antigens. This is a little bit different in the sense that it only is looking at the very early events with regard to response to a signal, a stimulation signal. And finally, through some complex systems, you can look at cell cycle change, again, which would be a measure of DNA proliferation. I already mentioned that the one effector assay that we typically can set up would be cytotoxicity. Either you measure chromium release from target cells that are preloaded with chromium-51, or there are flow cytometric methods in which you evaluate the expression of CD107A on the actual cytotoxic effector cell. This is actually generated or moves to the surface of the cell after the cell engages the target and develops the cytotoxic synapse. There are really two general categories of cytotoxicity. Antigen-specific, which is a T-cell mediated process, requires pre-sensitization and is initiated through the T-cell receptor. A classic example would be T-cell directed cytotoxicity against influenza-infected or EBV-infected target cells. It's exclusively specific to the particular antigen expression. In this case, these would be virally induced antigens expressed on the surface of the target cell. It also requires MHC compatibility because it is seeing antigen in the context of MHC. In this case, because it is affected primarily by CD8 T cells, it would be a class 1, HLA A and B dependent compatibility situation. Non-antigen specific is mediated primarily by NK cells that recognize classic and non-classic MHC class 1. I hope you'll hear a little more about NK cells. They actually have both an inhibitory and a facilitatory system that distinguishes self from non-self. And so these are ready activated. That is to say, they do not require pre-sensitization. They're armed and ready to go. And if you think about it, you don't want them indiscriminately knocking off your cells. So they know how to tell your own cells from cells that fail to express class 1, like tumor cells. And then there's something called antibody-dependent cellular cytotoxicity, or ADCC, which is actually mediated through bound antibody, that is typically IgG that coats target cells, and then FC receptors on NK cells, on monocytes, on macrophages can actually mediate cytotoxicity through this bridge between the target cell, the immunoglobulin on the target cell, bringing the target cell to the effector cell through the Fc gamma receptor. Okay, enough on function. So we're going to turn to innate immunity. All right, so we've done briefly B-cell antibody-mediated immunity. We've done T-cell or what historically has been called cellular immunity, but of course innate immunity is mediated in many cases by cells. So I think cellular immunity is much better replaced by T-cell mediated immunity. And so there are different aspects, and one of the new areas of immunodeficiency is really focused on toll-like receptor function. Keeping in mind, you guys are youngsters. When I was and Jay were a bit younger, maybe a little less gray, the TLR system was actually not even appreciated. This is a product of the late 90s, significant enough that the Nobel Prize was awarded to 3 investigators who, one of whom in Germany initially described the toll-like receptor on the fruit fly, and then 2 additional investigators who brought this to humans. We now have at least 10 TLRs on humans. And so the TLRs are pattern recognition receptors. They identify patterns that are products of non-mammalian cells, that is, materials that we do not make, do not synthesize, do not produce. So these are foreign products. It is an extraordinary system of early response, primarily directed against products of either extracellular pathogens like lipopolysaccharide or endotoxin, which activates TLR4, or intracellular foreign products that are produced by viral infections. So there are TLRs that are both extracellular responders, and there are intracellular or lysosomal TLRs that respond to viral products. And so there are distinctive clinical phenotypes associated with TLR dysfunction, one of which is recurrent pyogenic infections with limited systemic symptoms, little or no fever, normal or very minimal increase in CRP, etc. These are all disorders that have been described during this millennium. These are disorders that have been only described since the 2000 period. The other classic phenotype for a TLR defect is recurrent herpes simplex encephalitis. So it turns out that they probably have also susceptibility to some other viral infections. Now typically it's much more difficult, even in today's world, to get TLR function done. There are a few commercial labs that do this, and let me just show you what's involved. So here is an assay for TLR function. You simply separate mononuclear cells, and then you stimulate with one of a variety of different ligands that activates one or more toll-like receptor. And I've already noted the one classic example, and that would be endotoxin, which stimulates TLR4. You culture the cells in the presence of the ligand, go for a fixed period of time, and then harvest supernatant and measure cytokine production. So it's a moderately involved assay. There is an alternative approach described by the Casanova group some years back, about 6 years ago, looking at the shedding of a particular surface protein from cells. It's a protein called CD62 ligand. It's a selectin. I have to be honest, we couldn't get the assay to work. So it sounded like a terrific screening test, but for us it didn't prove to be terribly useful. Right. So within the context of innate immunity, we have the TLR function, and trust me, there are other disorders that will or already are being described. There are the production of local antibiotics that will most assuredly be identified as defects in the innate immune system. But remember, neutrophils are part of the innate immune system, and we've dealt with neutrophil deficiencies for more than 40 years. And so let's quick go through what we think about when we talk about screening for neutrophil function. I think you're all well aware that in this case we have a very classic phenotype clinically. Recurrent bacterial and fungal infections that typically involve skin, lungs, liver, bone. So there are 3 general categories. One is neutropenia. We now know there are a host of congenital defects associated with neutropenia, and then there's also iatrogenic neutropenia. We do it with drugs. But this has become less and less of a problem because we have GCSF. So patients with persistent iatrogenic neutropenia are far less common than they were 20, 30 years ago. In terms of genetic defects, there are really just, at this juncture, two major categories that are associated with significant clinical infection, significant morbidity, and significant mortality. One is a defect in oxidative burst, chronic granulomatous disease. I'll show you an example of a test that we actually pioneered, the dihydrorhodamine test, looking at oxidative burst. The NBT test is a much older nitroblue tetrazolium assay, which is a slide test, still done in a few institutions. Unfortunately, there are few people that really know how to read them effectively. It's not used much. And so although it's a simple test, it's not done very well in most institutions. You don't have that. We have about 300 CGD patients. Most institutions have half a dozen. And so we really think the DHR test is the right way to go. I don't think anybody does chemiluminescence anymore.
No, we've diagnosed 3 in the last 6 months. So we've had a run on CGD patients. We used to do the NBT test. I remember doing it, but now we do the DHR.
Yeah, it's a slam dunk test, although I have to say that we found some of the commercial labs have some problems, and I'm going to have a comment about that. And the other congenital disorder is one of adhesion molecules. The classic disorder that we see in this country is leukocyte adhesion deficiency type 1, or LAD1. There are other defects, but they are primarily in populations of consanguineous marriages, and really LAD2 and 3 are incredibly uncommon. LAD1 is relatively uncommon, but there are a number of patients in the U.S., and that's a defect in CD18 expression, which results in a failure to express beta-2 integrins. You can test for CD18. You can also test for CD11A, B, and C. When CD18 isn't produced, CD11A, B, and C are also not expressed on the surface of the neutrophil. So let's go through the two assays. Here is a DHR assay of oxidative burst. You load neutrophils with DHR, stimulate cells with a very potent stimulus called 4-ball myristate acetate. And if you see the normal up here on the left, you don't have to be a very sophisticated flow cytometrist to see that in the presence of PMA, there's a big change in terms of fluorescence. And this is a log scale, so this is almost 200-fold increase. Again, you don't have to be too sophisticated to say, hey, this is really different. And this is the mom. Mom has two populations. She's got a normal X chromosome. These are all the granulocytes that carry the normal X chromosome. These are all the granulocytes that carry the abnormal X chromosome. That's called lionization. In all females, only one of their two X chromosomes is active in every cell. And here you see a distribution where it's a random process of inactivation. So she has a mixed population of normals and abnormals. I can tell you that the distribution, although it's supposed to be 50-50, it's hugely variable in carriers. We've seen as low as 10 or 12% normal and as low as 10 or 12% abnormal. When you start getting around 10% or below, then moms actually can be symptomatic. Not as severe as a full-blown CGD, but they will, by history, have some infectious problems. This is the classic autosomal recessive patient. They have a little bit of oxidase activity. Remember, this is a log scale, so this is probably about 5 or 6% of normal. It looks much more impressive because of the log scale. Remember, this is 10-fold, this is 100-fold brighter than this. It's also classically a broader peak, very different than here. Suffice it to say that a small subpopulation of X-linked CGD patients look like this. So in our mind, this is quite diagnostic of X-linked CGD, but this does not discriminate between autosomal recessive and the less common X-linked CGD. Our group published, namely the CGD people here, data that patients that have some oxidase activity in the DHR assay, this was in the New England Journal in December 2010, actually have much less morbidity than the patients that have essentially no oxidase activity. And I'd refer you to that paper. I think Doug Kuhns was the first author, Kuhns, in the New England Journal in December of 2010.
So even a little bit of function helps.
And even a little bit of function helps. And that may be of some utility in making decisions about bone marrow transplant. So here is a classic LAD patient, CD18 expression. Again, it's a log scale. Control, patient. Patient has no CD18. It doesn't have absence. This is less than 10%. It's about 7%. Some LAD1 patients have zero CD18. Those are patients that end up going to transplant in infancy. They are sick as can be early in life. There's an important finding here, and that is using PMA and looking 15 minutes later, see what happens to the normal. They increase their CD18 almost threefold. Kind of makes sense. You go to an inflammatory milieu, your neutrophils have CD18 sitting ready to be expressed. This is not new protein synthesis, making them even more effective in being able to diapedese, to cross through the wall of a blood vessel to get to a site of inflammation. Look at the LAD patient, zero increase. So you go from 7 or 8% of normal down to 2% of normal. So what you can see is their disadvantage is actually magnified at a time of inflammation, explaining why they really have trouble getting their neutrophils to sites of inflammation. And so one of the classic findings in LAD1 is a marked increase in circulating neutrophils. They're stuck in the blood, in the circulation. They can't get out because of the absence of beta-2 integrins. I don't talk a lot about complement. I don't know much about complement. But there are two general categories of complement deficiency. Recurrent encapsulated bacterial infections, very much like antibody deficiency. These are early component deficiencies, and in most cases are associated with a substantially increased risk of autoimmunity, particularly SLE. These guys have very high incidence of SLE. In fact, C1q is like 90%. They're rare as hen's teeth, but just keep in mind, this is infection and autoimmunity. Now, that's a theme. I just wrote a chapter for an autoimmunity text about autoimmunity and immunodeficiency. And in fact, there are a host of different immunodeficiencies with a substantial risk for certain types of autoimmunity, pointing out that many immunodeficiencies are more than just defects in host defense. They're also immune dysregulation states with risk for autoimmunity. The other general category of complement deficiency is recurrent meningococcal infections associated with autosomal recessive deficiencies of the terminal complement component, C5 through C9. And actually, I think anybody that has 2 episodes of meningococcal meningitis must have a CH50. You can actually argue that it's a cheap enough test that one case of isolated meningococcal meningitis deserves a CH50. Why? Their siblings have a 1 in 4 chance of inheriting a defect if they actually have a terminal component deficiency. There are different papers with different incidents of terminal component deficiency in the context of meningococcal meningitis, as high as 14% in one series, as low as less than 1% in another series. Again, non-epidemic meningococcal meningitis. I'll leave it to Jay to say, I personally think doing a CH50, which is a relatively cheap test, is very useful. It tunes you into whether or not the patient is at risk for a recurrent episode and whether siblings, who also then would be screened if the patient is positive, should be carrying a medic alert badge or bracelet that they are at risk.
Yeah, our recommendation is that you do it after the first infection because it tells you whether they're at increased risk of getting another one.
Right, absolutely. If you need specific component testing, you really need to go to specialized labs. The one lab, I have no stake in it, is Patsy Gickliff's lab at National Jewish. It's the only lab in the United States that really does thorough complement evaluation, and she is spectacular as long as she continues to work. She's talking about retirement. I have no idea what will happen when she retires. All right, we're going to launch into molecular testing. I started 5 minutes late. I'll make Phil wait 5 minutes. I think these are important concepts for you to know. Newborn screening in particular, a little about T-cell spectrotyping so you at least know what it is, and 3 slides on mutation analysis, which is fundamentally ridiculous to try to cover in 3 slides, but we'll do it anyways. All right. So what you have to know, I've already talked about the fact that in order to generate a T-cell antigen receptor, you have to go through a somatic recombination process. This is a simplified diagram of what happens to generate a T-cell antigen receptor. So you have to loop out pieces of DNA to bring the V, the D, and the J together. And what you see is that you lose elements when you loop out. And here is what is called an episomal DNA circle. So when you produce a mature T-cell antigen receptor in the thymus, that process results in this piece of DNA that is no longer a nuclear DNA component, is not replicated when cells divide. So what we know is the T-cell receptor excision circle, or TREC, is generated in the thymus during T-cell receptor rearrangement, and that the cells that have this particular episomal DNA are naive T-cells. They've just gone through their T-cell antigen production. They're naive cells, and when they leave the thymus, they contain TRECs. If they encounter antigen and undergo a series of divisions, which is what happens when antigen stimulates T-cells, the daughter cells generated during this antigen-stimulated proliferation no longer contain TRECs. So there's a dilution of the TRECs that's magnified quite substantially. And so Jennifer Puck, some years ago, came up with the concept. So TRECs were used originally primarily in the HIV world, looking for evidence of thymic reconstitution using a host of different stimuli. She actually theorized that if one could take the Guthrie card and take a punch from that blood, that spot of blood taken from the heel stick, and extract DNA, that one would expect, since virtually every T-cell in a newborn is a naive T-cell. Obviously, they've come from a sterile environment, in utero, unless they've been exposed to an in uterine infection. And so one would envision that essentially all of their circulating T-cells are naive T-cells. And this has been borne out looking at cord blood and using flow cytometric methods such as CD45RA, CD62 ligand expression, that T-cells are indeed naive in cord blood. So they actually proved that if you take a small punch, extract DNA, and then go ahead and do a PCR assay for TRECs, that there are plenty of TRECs in cord blood, in blood obtained from a newborn nursery, and in blood obtained from a special intensive care nursery. If you do the same from samples obtained from SCID patients, either autosomal recessive or X-linked SCID, this is the initial data, it's been replicated, and I'll comment further, essentially no TRECs. This has now been actually institutionalized, and the Department of Health and Human Services is now recommending that all states add the TREC assay to their newborn screening. So I was recently at a meeting in which data was summarized from 5 states, involved screening of over 1.3 million newborns. The states are Wisconsin, the first state to screen, California, New York, Massachusetts, and Connecticut. Louisiana started it. They ran out of money, stopped. There are a number of other states that are online to begin screening. We now know what the incidence of SCID is, at least based on the first 1.3 million. It's about 1 in 60,000 live births. This has been borne out in all states. We also know that this detects complete DiGeorge syndrome. And interestingly, there's a new entity, lymphopenia of unknown origin in the newborn period. We have no idea what that is. And it's an interesting and, of course, unintended consequence of this screening. Hopefully, we'll know the etiology, or at least know. So we know there is hypogam, transient hypogam of the newborn period. And perhaps there's going to be something like transient lymphopenia of the newborn period. The TREC assay has proven to be less reliable in premature infants. Everybody has found that prior to 37 weeks of estimated gestational age, there's a much higher frequency of false positive TREC assays. And so in most states, they do either repeat assays or wait until 37 weeks of gestational age before they consider the assay reliable. Follow-up in these 1.3 million, after one repeats the TREC to confirm that it is indeed extremely low or absent, it then moves on to doing flow cytometry, looking at the peripheral lymphocytes and obtaining an immunology consult. In about half of the abnormal TREC assays, this resolves the abnormality. That is to say, it is no longer an abnormal TREC. Remembering the incidence of abnormal TRECs is extraordinarily low. And so it turns out it is a very cost-effective test. It costs about $5 to $6 to do a TREC assay. Okay, changing gears. We've talked about the T-cell antigen receptor. I hope you all realize that we have literally millions of different T-cell antigen receptors. We have a capacity to look at diversity within T-cell antigen receptors using a PCR-based assay called spectrotyping or immunoscope. And here is data from a normal. And all we're doing is really looking at different size products for different V beta families, V beta 1, V beta 2, etc. So what I can tell you about each of these products is each one of them differs by 3 nucleotides. I hope you realize that 3 nucleotides code for 1 amino acid. So in order to generate a real receptor, each of these, this would be the shortest receptor, 1 amino acid less than this receptor, 2 amino acid less than this, 3, etc., etc. It's a Gaussian distribution. Within each of these peaks, there are actually multiple different T-cell antigen receptors. All we can say is they have the same size. But what you get is a feel for diversity here. Each of these is diverse. It just so happens in this normal, V beta 23 is just actually not represented. And that's sort of typical of normals, that there may be 1 or 2 V beta families not. Here is a clonal receptor. It's actually a T-cell line from a tumor, which tells us we can see clones associated with malignancy, but we can also identify T-cell developmental defects. Here is a complete DiGeorge syndrome. Here is normal for these 3 V beta families. There are instances in the SCID spectrum where this assay is quite helpful. Omen syndrome, which is an incomplete SCID, is associated with oligoclonality. This is oligoclonality, 2 or 3 peaks. Omen syndrome has all of the disadvantages of SCID plus a lot of autoimmunity related to now at least 7 different defects. The original defects were RAG1 and RAG2, in which they generate T cells of a very limited clonal nature, most of which are autoreactive, actually to the host disadvantage. They provide no host defense, but they actually are autoreactive. So you have the same sort of infectious risk that a full-blown SCID patient has, with the added disadvantage of autoreactive cells that go attack skin, may attack liver, etc. So it's really a bad situation, a situation that requires the same speed with which one moves to bone marrow transplant. There's an analogous situation in the DeGeorge syndrome, which is called atypical complete DeGeorge syndrome, in which autoreactivity is the same pattern as seen in DeGeorge syndrome, again with an increased risk for serious opportunistic infection. So let's talk about sequencing in the last 3 minutes. So here is Sanger sequencing. We simply use a mix of what we call deoxynucleotides. Those are the building blocks of DNA. We polymerize using DNA polymerase. So we're simply building DNA using a template. We add, in addition, dideoxy. You see here, DDNTP, dideoxynucleotide, of the 4 building blocks. Remember, DNA has a very short alphabet, only 4 letters. We use 4 different dideoxy, each with a different color. These are what we call chain terminators. So when they're incorporated, the DNA stops. You stop building DNA. And so what you see is that you have this random mix of DNA, that each of which is, or they're different lengths, and the final incorporated nucleotide has a color. And so each of these, you can see here it incorporated a T, here incorporated a G, here incorporated an A, C, T, G. So you can actually build the sequence based on the color and the length. And the length is determined using an automated capillary electrophoresis system. And so this is what you get. You get a printout of nucleotides in sequence. And I picked the simplest of all worlds, X-linked SCID, and you can see here is the patient with a red nucleotide, here is the control with a blue, and here is mom with red and blue because she has 2 X chromosomes. So this is Sanger sequencing. Well, what's the downside? It's expensive. You have to do gene by gene. You have to find a lab that does it. Typically, you start with genomic, that is just DNA extracted from cells. And there are a lot of times when we get into issues that we can't answer with genomic DNA, then we have to go to complementary DNA, which is actually DNA synthesized from messenger RNA. We may have to look at message, protein, and function. The whole ball of wax. Sometimes we do 7 different genes to try to rule out a particular disease complex. If you're paying for that, that can be anywhere up to $2,000 a gene. We're a funded federal lab. Where are we going? Next-gen sequencing. It'll be with us in a clinical setting within the next year or 2. Don't really have time to talk about it. I want to just bring you up to date on one concept that is absolutely crucial that you understand, and that's the fact that we all have polymorphisms within our genes. The most common are SNPs, single nucleotide polymorphisms. These are single nucleotide differences that are present in greater than or equal to 1% of the population. This is ethnically defined, so African Americans have different SNPs than Caucasians, than Asians, etc. They occur approximately 1 in every 1,000 bases, but it isn't 1 in every 1,000. It's just that there's about 3 million SNPs. This is probably not so critical. They can occur in both coding areas and non-coding areas. Why is this important? If you detect a single nucleotide change in a patient, you better make sure first that this is just not a polymorphism, i.e. not a disease-causing gene. There's all sorts of databases. They're predominantly right now from Caucasians, although it's being built up in African Americans, it's being built in Africans, and it's being built in Asians. So this is a change that would not be a disease-causing chain, or not necessarily a disease-causing chain, because we know, for instance, SCID is not seen in 1% of the general population. So let's just close with what's on the horizon. I would say there are new approaches to immune testing, particularly in the innate world. Whole exome or genome sequencing will be readily available in the near future. They're already talking about the $1,000 whole genome. That would be cheaper than going after a single gene. What do we do with 3 billion pieces of data on each individual? This is a computer nightmare. And so the challenge of genomics will be how to interpret and manage this massive amount of information. In your lifetime in medicine, it is going to become a part of the general evaluation of patients. I'm old enough that I'm not entirely sure it will be clinically useful in my short remaining career. And with that, I will close out this part of your morning lecture, and I only went 5 minutes over, Jay.
Okay. Well, thank you so much for that presentation. That was amazing. Normally, I would like to have questions and answers, but I think given the
email questions if they like.
Yeah, why don't if you have questions for Dr. Fleisher, feel free to email him. We'll post the email address on our website.
Okay.
But thank you so much. That's
all right,
been
Jay. Take
very
care.
Interesting, and we really appreciate it.
All righty.
This has been an ACAAI production. To learn more about conferences on line allergy or the American College of Allergy, Asthma, and Immunology, go to acaai.org. See you next time.